Today we're going on a deep dive into some really fascinating science, a breakthrough, really. There's a recent study, it was covered in Newsweek, based on research in Science Translational Medicine. It's about a new HIV vaccine strategy. It comes from researchers at MIT and Scripps Research Center, and people are already calling it Supercharged. So our mission today for this deep dive is to unpack what exactly makes this HIV vaccine so, well, supercharged. We want to explore its unique design, how it's supposed to work inside your body, like at the cellular level. And really importantly, what are the bigger implications? Not just for HIV, right? But maybe for other vaccines too. Things like the flu, maybe even future pandemics. We really want you, our listener, to walk away feeling like you get it. Not just the headlines, but the real how and why behind this cutting edge research.
And that's exactly the plan. It's not just about telling you what this is. We want to get into how it works, the mechanics of it, and maybe more importantly, why it matters for you to understand this science. Because this isn't just, potentially one new vaccine. It's about a whole new approach, potentially. One that could really change how we think about making vaccines for all sorts of infectious diseases, maybe more efficient, more powerful ways than we thought possible for some of these really tough bugs. So for you, the listener, it's about really grasping the science underneath it all, moving past the buzzwords, and seeing why it could be so significant for public health across the globe.
Right. And breaking it down is key because, the implications could be huge. Let's start with that headline then. It's pretty exciting just on its own. Supercharged vaccine could protect well with just one dose. That's a really bold statement for any vaccine, wouldn't you say? Especially for HIV. I mean, this virus has been incredibly tricky to develop a vaccine for. Decades of effort.
Oh, it's absolutely a remarkable claim. You really have to appreciate the context of HIV vaccine research to see how big a deal this sounds.
Yeah.
HIV is it's a formidable opponent for the immune system. It replicates incredibly fast, which means it mutates constantly. Just constantly changing. So its outer surface, the part the immune system usually targets, it keeps shifting. It's like trying to hit a moving target that's also changing colors. Past attempts, you might remember some big trials like the STEP trial, they often struggled because they just couldn't provoke an immune response that was broad enough or strong enough to handle all that variation.
Right, it's like the virus is always one step ahead.
Exactly. So when top researchers start talking about single dose, strong protection for HIV, everyone in the field sits up and pays attention. Now, these initial studies, we should be clear, they were done in mice. But they did indicate this vaccine could potentially offer really robust protection with just that single shot. And the impact of a single dose of this vaccine, think about it globally. For HIV, it's almost hard to overstate. Think about the logistics. Multi-dose schedules are tough, especially in places with limited resources or just for getting huge populations vaccinated quickly.
Yeah, getting people back for second or third shots is always a challenge.
Always. A single shot just cuts through all of that. Better compliance, simpler distribution, faster protection for communities. It changes the whole game. And the fact that this research comes from places like MIT, the Massachusetts Institute of Technology, and Scripps Research Center, well, that adds serious weight. These aren't fly-by-night operations. You're looking at rigorous, high-caliber science. Their involvement really tells you this is something worth looking at closely.
Okay, so single dose, supercharged. That's the promise. But when I hear supercharged, my first thought is this some brand new, untested kind of technology, like something totally out of left field? Or is it more about using things we already kind of understand, but in a new, maybe smarter way? What's really driving this effect?
That's a really good question. And actually, it's more of the second thing, which is part of why it's potentially so exciting. It's not based on some completely alien tech.
Yeah.
The secret sauce, if you want to call it that, comes down to something called adjuvants.
Adjuvants, okay.
Yeah. Adjuvants are basically ingredients that get added to vaccines. They aren't the main target themselves. That's the antigen, the piece of the virus or bacteria your body needs to recognize. Adjuvants are more like partners. Their whole job is to boost your immune system's response to that antigen.
Like hype men for the immune system.
Yeah, that's actually not a bad way to put it. Or maybe personal trainers. They make sure the immune system really notices the antigen and mounts a stronger, better response than it might otherwise. And adjuvants themselves, they're not new at all. You've likely encountered them before. Probably the most common one is alum. It's short for aluminum hydroxide. It's been used for decades in lots of protein-based vaccines. Things like hepatitis A, hepatitis B vaccines often contain alum.
Okay, so it's a known quantity.
Very much so. It's kind of the classic immune booster. What's interesting is that most vaccines you get today already use some kind of adjuvant to work better. So the concept is standard practice. Alum, for instance, it basically works by ringing an alarm bell. It wakes up your body's first line of defense, the innate immune system. Think of them as the security guards who notice something's up. That initial alert helps your body build a stronger, more lasting memory of the actual vaccine antigen. It lays the groundwork. And, the history is pretty long here. Alum's been used since, I think, the 1920s. So it has a very long track record, which is reassuring. But science is always looking for better ways, right? Especially for tough targets like HIV, where the standard methods haven't quite cracked it.
So adjuvants are the trainals, but this new vaccine isn't just using the standard trainer, is it? You said it's a combination. How did they make the training program, more intense, more effective?
Exactly. It is a combination. Yeah. And that's where the real cleverness, the innovation comes in. This HIV vaccine isn't just using an adjuvant, it's using two together in a very specific, strategic way. And it seems like the synergy, the way they work together, is what creates that supercharged effect we're talking about.
Okay, 2 adjuvants. So what are they?
Well, one half of the team is the familiar alum. Provides that basic boost, gets the innate system going like we discussed. But the second part, that's the new player here. It's a novel nanoparticle called SMNP.
SMNP. Okay, what's in that?
So this SMNP nanoparticle, it's pretty cleverly designed. It actually combines 2 things itself. One is a substance called saponin. Now, saponin is naturally derived, and importantly, certain forms of it are already FDA approved for use in other vaccines. So again, there's some familiarity and a known safety profile there, which is helpful.
Right, not starting completely from scratch.
Exactly. That potentially helps smooth the path towards, eventual human trials down the road. Saponin is another type of adjuvant itself. And the SMNP combines this saponin with an inflammation-promoting molecule.
Inflammation-promoting? That sounds potentially bad, or is it strategic?
It's very strategic in this context. A little bit of controlled, localized inflammation is actually part of how adjuvants work. It signals danger to the immune system and helps attract more immune cells to the injection site, or in this case, towards the lymph nodes where the real action happens. So this molecule helps amplify the signal, like putting up more posters to recruit immune cells to the training academy. And what the researchers found in their early experiments was pretty striking. This dual adjuvant vaccine, the alum plus the SMNP, it produced a significantly wider diversity of antibodies against the HIV protein they were testing compared to using just alum alone or just SMNP alone or no adjuvant at all.
Diversity. Okay, let's unpack that word, because for HIV, that sounds incredibly important. You mentioned earlier how much HIV mutates. Is this diversity like building up a whole arsenal of different weapons instead of just one type?
That's an excellent analogy, yes.
Yeah.
That diversity isn't just a minor detail. It's potentially the absolute key. For decades, like you said, HIV has basically outsmarted our immune systems and our vaccine attempts precisely because it's a master of disguise. It mutates so fast. So if a vaccine teaches your body to make just one specific type of antibody, the virus can just change that little part of itself and poof, the antibody doesn't work anymore.
Like changing the lock when you only have one key.
Precisely. So what this diverse arsenal of antibodies suggests, it's like building, as you put it, maybe a whole SWAT team. Or maybe it's like giving your immune system a master key ring with lots of different keys. Each antibody, or family of antibodies, might be slightly different, potentially able to recognize different versions, different variants of that HIV protein. This means the immune system isn't just learning one solution to the problem, it's learning multiple solutions. That massively increases the odds that if you encounter the real virus later, your immune system will have something in its toolkit that can recognize and neutralize it, even if the virus has mutated somewhat.
That feels like a fundamental shift. Instead of trying to find the one magic bullet, you're training the immune system to be more adaptable, more flexible.
Exactly. It's preparing for the variability rather than being surprised by it. It's a proactive strategy against a constantly shape-shifting enemy. That's why the diversity finding is so exciting here.
Okay, so this dual adjuvant team sets the stage for a more diverse response. Let's get into the mechanics then. How does this actually happen inside the body? You mentioned lymph nodes earlier. Are they the key location for this supercharged training?
Absolutely. Lymph nodes are central to this whole process. You can think of them as, well, strategic command centers is one way, or maybe specialized training academies for your immune system.
Right, the boot camp for immune cells.
Pretty much. This is where key immune cells, especially these crucial white blood cells called B cells, congregate. And it's inside the lymph nodes that these B cells get properly introduced to antigens, those bits of the virus the vaccine presents. They don't just meet the antigen, they undergo this intense training process. It's called affinity maturation. Basically, they learn to produce better and better antibodies that bind really tightly and effectively to the antigen. So yeah, the lymph nodes are where the critical learning and weapon making happens for a long-term antibody response. You need good training in the lymph nodes for a good vaccine.
And what did these researchers find out about how their specific combo, the alum plus the SMNP, changes what happens inside those lymph nodes?
This is where it gets really, really interesting. They uncovered something quite remarkable about how this combination affects the antigen itself. They found it achieved two really vital things in the mice studies, things that don't typically happen with standard vaccines. First, the combination somehow helped the HIV protein they were using as the antigen. It helped it get into the lymph nodes more effectively. It seems to help it penetrate that outer layer of cells around the lymph node structure. And critically, it did this without the antigen being broken down or degraded too quickly.
That sounds important. You need the training material to stay intact, right?
Exactly. If the antigen gets chewed up too fast or can't even get to the right classroom in the lymph node, the B cells don't get a good look at it. The training is compromised. Imagine trying to learn from a textbook that's, dissolving as you read it. So getting the antigen into the lymph node intact is step one. But step two was perhaps even more crucial. They discovered that this specific dual adjuvant combination allowed that intact antigen to actually stay inside the lymph nodes, concentrated, for a really extended period. We're talking up to 28 days.
Wait, say that again? 28 days?
Up to 28 days, yeah.
Wow, that's nearly a month. How does that compare to, like, a standard vaccine?
It's a huge difference. With most typical protein vaccines, the antigen is cleared out pretty quickly, maybe hours, maybe a couple of days at most. The immune system gets a relatively brief exposure, so keeping it there, available for interaction for up to 4 weeks. That's a fundamentally different scenario for immune learning.
It's like the difference between a quick seminar and a month-long immersive course.
That's a perfect analogy. And Professor J. Christopher Love, he's one of the senior authors, an MIT chemical engineer, he really emphasized this exact point. He explained it by saying basically, as a result, the B cells that are cycling in the lymph nodes are constantly being exposed to the antigen over that up to 28-day time period, and they get the chance to refine their solution to the antigen.
Refine their solution. I like that phrasing. It sounds like an iterative process, like they keep trying and getting better.
Precisely. It's not just about making an antibody. It's about making the best possible antibody. Inside specialized areas of the lymph node called germinal centers, B cells go through this incredible process. They multiply rapidly, they introduce tiny mutations into their antibody genes, and then they get tested. Only the B cells whose mutated antibodies bind best to the antigen get signals to survive and multiply further. The ones that don't bind well, they basically get eliminated. It's like natural selection happening at hyperspeed within your lymph nodes.
Right.
So having the antigen hang around for 28 days gives this refinement process much more time to happen. More rounds of mutation, testing, and selection. It allows the immune system to explore many more possibilities and ultimately select for those B cells, producing truly high-affinity, potent antibodies. Professor Love also added, when you think about the immune system sampling all of the possible solutions, the more chances we give it to identify an effective solution, the better. It just underscores how important that extended exposure time is for the quality of the final antibody response.
Okay, so the theory makes sense. Longer exposure, better refinement, higher quality antibodies. What did they actually see when they looked at the B cells and antibodies produced in the mice that got this dual adjuvant vaccine? Did it translate?
It absolutely did translate. They performed RNA analysis on the B cells, which gives you a really detailed picture of which B cell types are present and how diverse they are. And the results were really clear. The mice that received both adjuvants, the alum, and the SMNP, they produced a significantly more diverse collection of B cells and consequently antibodies compared to the mice that got only one adjuvant or no adjuvant at all.
How much more diverse are we talking?
Quantitatively, they found that the mice getting the dual adjuvant vaccine produced more than twice as many unique B cell lineages compared to mice who received a vaccine with just a single adjuvant.
More than double the diversity. That's not a small difference.
Not at all. It's a substantial increase. Again, bringing it back to HIV, this is exactly what you want to see. That increased diversity directly translates into an enhanced ability to potentially recognize and neutralize a wider variety of HIV strains. Given HIV's notorious ability to mutate and exist as countless different variants or strains across the globe, and even within a single infected person, having that broad, diverse antibody arsenal is absolutely critical. It addresses one of the fundamental reasons why previous HIV vaccine candidates have often failed. They generated responses that were just too narrow, too specific, and the virus could easily escape. This approach seems to tackle that head-on by driving breadth right from the start.
So it's not just a stronger response, it's a broader response, like having a tool for every possible variation the virus might throw at you.
That's the goal, yes. A more adaptable, resilient immune defense.
Okay, this is clearly huge for HIV, but you hinted earlier, this isn't necessarily just about HIV, is it? Could this dual adjuvant, long exposure strategy be applied to other diseases too?
That's definitely the hope and a major part of the excitement around this research. The researchers themselves, particularly Professor Love, seem quite optimistic about its broader potential. He was quoted saying something like, "This approach is compatible with many protein-based vaccines," which is a key phrase.
Protein-based vaccines, remind us what those are.
Right. So many vaccines work by using a specific protein, or sometimes just a piece of a protein, from the virus or bacterium as the antigen. Your body learns to recognize that protein. A lot of familiar vaccines fall into this category, things like the hepatitis B vaccine, the vaccine for diphtheria, tetanus, pertussis, DTaP, the HPV vaccine. They often use protein components.
Okay, so it's a common vaccine type.
Yes, a very common and well-established type. So when Professor Love says this dual adjuvant approach is compatible with protein-based vaccines, it means it could potentially be used to supercharge many existing vaccine types, or new ones being developed for other diseases that use protein antigens. You went on to say it offers the opportunity to engineer new formulations for these types of vaccines across a wide range of different diseases.
Did he give any examples?
He did. He specifically mentioned things like influenza the flu, which, as we know, also mutates, though not as dramatically as HIV, and SARS-CoV-2, the virus that causes COVID-19. And he also alluded to, other potential pandemic threats that might emerge in the future.
Wow. So this could be more like a platform technology, a new tool in the toolbox for making better vaccines in general.
That seems to be the implication, yes. Imagine if you could take a protein antigen for, say, a particularly nasty strain of flu or a new emerging virus, combine it with this SMNP alum adjuvant system, and potentially get a highly effective, broad, single-dose vaccine much faster than before. That would be a game changer for public health preparedness.
It really would. Okay, let's bring this back home for our listener then. We've gone through adjuvants, B cells, lymph nodes, 28-day antigen exposure. Why should someone listening right now really care about these, seemingly small details of immunology? What's the real world so what here?
The so what is potentially enormous. It really boils down to the promise of more effective, more convenient, and more accessible protection against major diseases. Let's think about that single-dose aspect again. If this pans out, if it proves effective and safe in humans, a single-shot vaccine strategy could just revolutionize global health efforts. First, compliance. We touched on this. It's just so much easier to get people fully vaccinated with one shot versus 2 or 3. Think about reaching everyone, busy schedules, access issues. A single dose overcomes so many barriers.
Makes total sense. Fewer drop-offs.
Exactly. Second, logistics. Mass vaccination campaigns are incredibly complex operations. Shipping, storing, especially if cold storage is needed, tracking doses, staffing clinics for multiple rounds. A single-dose regimen just simplifies everything dramatically. It makes campaigns cheaper, faster, and allows you to reach more people, especially in remote or resource-limited areas. Think about the impact for global equity and access to vaccines.
Right, gets protection out further and faster.
And third, and this feels increasingly important, is the speed of response. If this really is a platform technology, if we can plug in a new protein antigen from an emerging threat, combine it with these adjuvants, and quickly develop a single-dose vaccine, that could drastically shorten our response time to new outbreaks or pandemics. We saw with COVID-19 how crucial speed was. Having a tool like this could allow us to get ahead of the curve much more effectively next time. So it's about more powerful prevention tools that are easier to use, easier to distribute, and faster to deploy. And another key point Professor Love made, the power here comes from combining reasonably well-understood components. Alum is ancient in vaccine terms. Saponin adjuvants are also used and FDA-approved in some contexts. So it's not like they have to invent entirely new molecules with unknown safety profiles from scratch. They're cleverly combining existing types of building blocks that could potentially make the path through clinical trials and regulatory approval a bit smoother and faster than if it were based on something completely novel and untested.
That's a really important practical point. It grounds the innovation in things that are already somewhat familiar, potentially speeding things up. But speaking of clinical trials, this is still based on mouse studies, right? What needs to happen next to see if this actually works in people? What's the road ahead?
That's the crucial next question, absolutely. Moving from promising results in mice to something you or I could actually receive is a long, complex, and very carefully regulated process. The immediate next step typically would be to test this vaccine strategy in larger animal models. Often this involves non-human primates like monkeys, because their immune systems are much more similar to ours than mice are.
Okay, so testing in monkeys first. What are they looking for there?
They'd be looking to confirm the key findings. Do they see that prolonged antigen presence in the lymph nodes? Do they see that broad and diverse antibody response? Crucially, can those antibodies actually neutralize different strains of SIV, the monkey equivalent of HIV, or perhaps challenge viruses engineered to mimic HIV? And of course, safety is paramount at every stage. They'd be monitoring very closely for any adverse reactions or side effects in these larger animals.
Assuming the primate studies look good, what then?
Then comes the big leap into human clinical trials. And these happen in distinct phases. Phase 1 is usually small, maybe a few dozen healthy volunteers. The main goal here is safety checking for immediate side effects and figuring out the right dosage range. They'll also look at the initial immune response. If phase 1 looks good, they move to phase 2. This involves more people, perhaps several hundred. They continue to monitor safety very closely, but now they're also getting a much better idea of the immune response generated in humans. Does it look like the response seen in animals? Is it broad? Does it seem promising? And if phase 2 is successful, then you get to the huge phase 3 trials. These can involve thousands, sometimes tens of thousands of people, often across multiple countries. This is where you really test for efficacy. Does the vaccine actually protect people from infection or disease compared to a placebo? And you continue monitoring safety in a much larger population.
And each of these phases takes time, right? Years, potentially.
Exactly. Each phase takes time, careful planning, significant funding, ethical review, and regulatory oversight. There are no shortcuts when it comes to ensuring vaccines are both safe and effective. So while this research is incredibly exciting and provides a really strong scientific foundation and a clear direction, it's important for everyone listening to understand that we're still at the relatively early stages. A usable human vaccine based on this is likely still years away, assuming everything continues to go well. But the potential pathway it opens up, that's what's generating so much justifiable hope and excitement in the scientific community.
Okay, that really puts it into perspective. The science is amazing, the potential is huge, but the journey to a real-world vaccine is still a marathon, not a sprint. So wrapping up our deep dive today, we've explored this really fascinating new approach to an HIV vaccine coming out of MIT and Scripps. It uses this clever dual adjuvant system. The familiar alum teamed up with this novel SMNP nanoparticle. And together, they seem to create this supercharged effect.
And the heart of that effect, the core insight, is how it changes the game inside the lymph nodes. By keeping the vaccine antigen intact and present for that extended period up to 28 days, it gives the immune system, particularly the B cells, much more time to go through that crucial refinement process. The result, at least in the early studies, is a more diverse, broader, and hopefully more potent antibody response, potentially achievable with just a single dose. It's about giving the immune system the time it needs to learn properly.
Which leads us to a final thought, something for you, our listener, to maybe chew on after this. If this kind of approach does prove successful, if we can apply this supercharged single-dose strategy using these kinds of well-understood but smartly combined adjuvants, if we can apply that not just to HIV, but to flu, to COVID, to other diseases, how might that fundamentally change things? How might it reshape our whole approach to public health, to preventing disease, maybe even change our own personal relationship with vaccines and medicine in the years and decades ahead?
Yeah, think about those ripple effects. The sheer accessibility of single-dose vaccines globally, the improved compliance, making protection more reliable, our ability to react so much faster to new threats. Could advancements like this truly shift the balance, move us towards a world where widespread, effective prevention becomes much more the norm, even for diseases that have challenged us for so long? It's a powerful example of science taking on incredibly complex problems with innovative thinking, and it really offers a hopeful glimpse into what might